So now in section 13.4, we're talking about the regulation of RNA processing and translation in eukaryotes. For the longest time, we've emphasized that For the longest time, we've emphasized the For the longest time, we focused on For many years, we've focused on the effects of proteins on transcription initiation and how we could use transcription factors either to enhance the initiation step or to repress that initiation step. And certainly that's a very important point of control in all cells. However, over the last 25 years, we've started to elucidate other mechanisms by which cells can regulate proteins in the cell. These are post-transcriptional events, either at the level of RNA processing, at the levels of translation, or even post-translation. So, what are the benefits here? Well, the benefits of, for example, of alternatively splicing RNA is that a single gene can form multiple products. So we see this example highlighted in the fact that all cells have a common genome, but yet not a common proteome. How can that happen? How can two cells with the exact same set of genes ultimately produce different proteins from those genes? Alternative splicing. The other point is that by going post-transcriptional, we can have a faster reaction by controlling steps after the RNA transcript is made. So we've talked about this before, alternative splicing of your primary transcript. Here's a nice example. Here we have a primary transcript that has multiple exons and multiple introns. So now in this example, in a nerve cell, the region corresponding to number 4 is taken out, is cut out. And now our final transcript is 1, 2, 3, 5, 6, 7. So this is an intron, this is an intron, 4 is an intron, 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 intron. So we have 1, 2, 3, 4, 5, 6 introns at each position. Now in the same primary transcript in a muscle cell is spliced differently. Let me get out my eraser. In the case of a muscle cell, intron 4 is now an exon. Exon 3 in a nerve cell is now an intron in a muscle cell. So now we have regions 1, 2, 4, 5, 6, 7, rather than 1, 2, 3, 5, 6, 7. And so now the result of this is protein A is produced in the nerve cell, but protein B produced in a muscle cell. Same gene, same primary transcript, but ultimately different products that allow a nerve cell to function uniquely compared to that muscle cell. So the advantage here is multiple polypeptides can be derived from a single gene, thereby increasing the size of the proteome while minimizing the size of the genome. 25,000 genes, 100,000 proteins. It can't happen unless we can find different ways to read the information in the genome of different cell types. Now, interestingly, when we think of biological complexity, we can now begin to correlate that with sizes of genomes and proteomes. As we talked about, alternative splicing increases the size of the proteome without affecting the genome. As organisms become more complex, such as in the case of plants and animals, evolution has produced more complex proteomes. And what we see is this is a general trend as you go through all living things. So E. coli is a bacterium which is a fairly simple unicellular prokaryote. Its genome is about 4.2 million base pairs. There are approximately 4,000 genes. There is 0 alternative splicing. So now let's leave the prokaryotic world, enter the eukaryotic world. Saccharomyces cerevisiae, a single-celled yeast. 12 million base pairs in its genome, 6,000 genes. Less than 1% of its genes are alternatively spliced. Let's go up now to Homo sapiens. Fairly complex multicellular mammal. 3,000 million base pairs. 25,000 genes. 70% of genes alternatively spliced. And so during evolution, as organisms diverged and as they found new ways to regulate gene expression, this idea of alternative splicing appears to be a key difference between complex plant animals, like the flowering plant Arabidopsis, and those simple single-celled eukaryotes and prokaryotes, like yeast and bacteria. Let's talk about microRNAs. Man, we learned about a lot of RNAs this semester. Messenger RNA, ribosomal RNA, transfer RNA, small nuclear RNAs. Now here's another class, microRNAs. These are small RNA molecules which do not encode for protein. These are terminal, meaning after transcription, they are not then sent to the translation machinery to produce proteins. What they do, however, is they act as silencers for existing mRNAs in the cell. Again, higher order organisms, we find these a lot, animals and plants. We do find them in bacteria as well, so I don't want to say this is unique only to animals and plants, but this is important in what we call mRNA silencing. Well, how does this work? Well, here's my microRNA. Now remember, in eukaryotes, transcripts are often modified. This is no exception. We have a pre-microRNA that has the ability to fold onto itself, forming here this hairpin loop and establishing hydrogen bonds between complementary base pairs. There's a molecule in the eukaryote called dicer, which is going to process this pre-microRNA, cleaving it at specific positions, and releasing ultimately the mature microRNA from the rest of the complex. Now, once that happens, this microRNA is able to establish a complex called a silencing complex, RISC, RNA-induced silencing complex. Now here's why that's important. Because in this form, it's able to establish complementary base pairing with existing messenger RNA. We can see that here. Now the effects could be twofold. The mRNA can be targeted for degradation when there's highly established complementary base pairing here. So the mRNA never makes it to the translational machinery. The mRNA is degraded. Well, it's hard to make protein from an mRNA that's not intact. So that can be silencing RNA, siRNAs, that are specific, can carry out the degradation step. For these microRNAs that are not as complementary, they can still work by inhibiting translation, because the ribosome is going to be blocked by this complex, unable to move 5 to 3. Well, listen, if the ribosome can't translate, no protein results. So we can see that these small RNAs have a negative impact on protein expression. Whereas the previous sections in this chapter dealt with the impact on transcriptional expression, here we're talking about an impact on protein expression.